In this lecture, we will discuss about the construction of non-redundant datasets. Why do we need non-redundant datasets? What is the meaning of non-redundancy? How these datasets influence the prediction models or any large scale data analysis. So, in the last lecture, so do you remember what did we discuss in the last lecture? Mainly about hydrophobicity profiles, right. So, what is the hydrophobicity profile? It is a plot connecting the amino acid sequence versus hydrophobicity values. So, we have experimental hydrophobicity values for the 20 different amino acid residues, right. For each residue you assign the values and construct a plot. Which information we will get if we construct hydrophobicity plots?
So, like secondary structures.
Correct. You can see some sort of patterns or motifs, right. So, you can see any specific patterns, for example, in alpha helices, right, or beta strands, for example, alternating hydrophobicity or transmembrane segments dominated with the hydrophobic residues, right. We can see some profiles and we can see the patterns and relate these patterns to any specific structure or function, right. Then we discussed about ambiphasicity, right. What is ambiphasicity? Right, it is a periodicity of this polar and non-polar residues in any amino acid sequence. You can see some specific periodicity in the case of alpha helices or beta strands and so on. Then we discuss about patterns, right. Patterns are defined with some symbols, for example, square bracket, right. What is the meaning of square bracket?
Inclusive.
Right, you can include any amino acids which are given within the square bracket, right. For example, if you have a square bracket and IIT, so any residues within this group is allowed. If it is curly brackets, so the residues which are inside this bracket, for example, if it is D, the D is not allowed under particular position. So, likewise you can make any motifs or you can see any patterns. You can also search the database, right, for using the PAI or any reference search, right. You can see the whole uniprot sequences, whether you can identify any specific patterns. Okay, the patterns and motifs are important for several functions. Then we discussed about the profiles, right, position specific scoring matrices or position weight matrix, right. How to construct the position weight matrix or scoring matrix?
So, we take the frequency.
Take the frequency of residues. First we need the multiple sequence alignment. From the multiple sequence alignment we get the frequency, then we convert the frequency into this matrix, alignment matrix, right, by normalizing with the probability of residues, right. For example, in the case of nucleotides, right, or for example, the amino acids. In the case of nucleic acids, right, the probability is 0.25, the amino acids you can 0.05. So, you can use the pseudo counts and finally normalize, right, you will get the matrix. So, we discussed various applications, for example, the prediction algorithms and the binding sites, right, different types of functional important sites and so on. So, now if you do the analysis in bioinformatics, it is very important to construct data set, because data set plays an important role in the analysis as well as in the prediction. If you have very good data set, right, then you can the reliability of any prediction methods is very high, right. So, because the results are biased with the data set, right. In this case, it is very important to construct a data set which are not redundant with each other. In this case, what is the meaning of redundancy? What is the meaning of non-redundancy? So, for example, if the two protein sequences which are similar to each other, you can take any specific cutoff, for example, 70% or 80%. If it is 80% in the sequence A and the sequence B, right, 80% of the sequences are the same or similar. So, if you add up these sequences, this will introduce a bias of this sequence, because they are influenced with the data which we obtain, right, from these sequences. So, in this case we need to avoid bias, we need to construct a sequence, non-redundant sequences at any specific cutoff depending upon the problem we choose. If you have 100,000 sequences, right, we have more number of data, right, then we can decrease the cutoff, we can use 20% or 30%, right, so that we will get sufficient number of data for the analysis. If the initial data set is small, then we need to set the cutoff accordingly, so that you will get sufficient number of data for the analysis, because for the redundancy this will cause a bias. So, it is important to construct a data set of completely non redundant, for example anything with 40% and so. For example, here I get two sequences, right. This is one sequence, sequence 1, and here you go another sequence, sequence 2, right. If we take these two sequences, what is the amino acid composition for alanine? Total here 10 residues, here 10 residues, total 10 residues. How many alanines? Right, 1, 2, 3, 4. So, 4 by 20, this is equal to 0.20, right. In some cases, for example, if one of the sequences appear twice, that means we introduce a bias in this analysis. For example, this sequence appears twice. Now, totally how many residues? 30 residues, right. Out of 30 residues, how many alanines? It is 7. So, if you take the composition, now it is 0.23, this is over represented. If you have the second sequence twice, then what is average alanine? Totally 30 residues, 5 by 30 equal to 0.17, right. Likewise, if you include the same or similar sequences in your data set and you extract information, you can see a bias, right, that we introduce because of the same sequence or similar sequences, right, you are including in your data set. Hence, it is very important to construct data sets for any analysis which are non-redundant with each other. So, how to get this non-redundancy, how to construct non-redundant sequences. So, in bioinformatics there are various approaches, right. I will explain this one of two important approaches, and several software available to construct non-redundant data sets. One of the most popular software, that is called CD-HIT. CD-HIT refers to the cluster database and high identity with tolerance. So, what this program does? So, our aim is to get the non-redundant sequences. For example, if you have 1000 sequences or 10,000 sequences, you need to extract the non-redundant data from this initial data set, right. So, here you have to give these sequences an input, because in this case we need to remove redundancy from the sequence. So, it takes FASTA format, right. FASTA format we discussed earlier, right. It starts with the greater than symbol, right. So, it takes all the sequences as input and it gives a set of sequences which are non-redundant, right. That depends upon the user's choice. We can specify the cutoff, we can specify the word size, right. I will explain now, right. And this CD-HIT will treat all your sequences as input, and depending upon your convenience or your needs, right, it will give you the non-redundant data set. So, how it gives a sequence, right. It uses a clustering algorithm to eliminate the redundant sequences. It takes your sequences and makes some clusters, and based on the clustering it will take the non-redundant from picking up the sequences from each clusters. What is the use of this program? Why this CD-HIT is very popular? Because this program handles a huge data sets, right. Even if you have thousands of data, it can handle the huge data sets and give you the results very quickly. Then it is easy to download, and this CD-HIT you can get the results very quickly. So, we can use the CD-HIT to create non-redundant sequences at different sequence identities, for example 40%, 50%, 60% and so on. The disadvantage of CD-HIT is it has some limitations to restrict the sequence redundancy, either up to 30% or 40% depending upon the program you use online or you get the this downloadable version. So, that is only disadvantage. Other than that, if you want to get any specific sequence cutoff up to 30%, right, we can use CD-HIT and it is easy to use and get the results very quickly. Let us see how the CD-HIT works. So, it uses the greedy incremental algorithm to select the protein sequence sets. So, it takes some clustering technique, right, try to assign each sequence and make into particular clusters and then check any of these features, and using the features you try again and again and again, right. This is called a greedy algorithm, unless the deviation is very less. Even then it is not satisfied. So, look for the other better solutions again and again, right, and to find the most probable solution for getting your non-redundant sequences. So, how it does? It takes the sequences and we can specify the identity, for example 40%, and this specific threshold, if it is more than the threshold, for example 40%, right, that will be discarded. So, then it takes the longest sequences first and then proceed with the shortest ones. And what is the sequence identity? Right, we discussed earlier, sequence identity is the number of identical residues divided by the length of the shorter sequence. For example, if you have two sequences, right, this is sequence 1, then sequence 2, so what is the sequence identity? Right, this is same 2, 3, 4, 5, 6 by 10, this is equal to 60%. If the two sequences which are more than a particular threshold, for example 40%, right, will it consider both or will consider only one? Only one, right, because it is more than 40%. So, do not keep both, so they keep one and discard other one, right, this is how it works. So, if you take the explicit algorithm alignment, so which algorithm we use for sequence alignment? BLAST, right. So, BLAST is widely used. If you have several sequences, for example thousands of sequences, if you want to make the sequence alignment, complete sequence alignment, it is very time consuming, right, it takes a lot of time to make the alignment and find these identities or not. So, this implements an algorithm without aligning the complete sequences. So, it takes a specific set or specific subset and then see whether you can find any subset of residues which are same between two sequences. For example, if two sequences A and B, they are considered to be 90% sequence identity, there is at least one peptides, right, at least of 8 residues like decapeptides or 10 residues which are available both in query and the database. If you look into these two sequences which are identical or similar or the high identity, then you can see long stretch of residues which are similar to each other. For example, 100 residues and the identity is 90%. How many residues are same? 90 residues are same. In this case, if you look in the sequence, at least some segments, if you see the large stretch of residues which are the same, based on that assumption, they try to use only decapeptides, also the different lengths, and see whether any segments, right, which are same in both the sequences. Then do not have to do the complete alignment, look for the stretch of residues. If they are same, then they assume that they are 90% identity. See, this is because of the reason. Why? Because it is highly redundant, there should be some stretches which are the same among different sequences. Then with different sequence identities, for example 85% or 80% or 75%, they try to reduce the length. For example 4 residues or 5 residues like pentapeptides, right, or tetrapeptides or tripeptides, and they also set more number of segments. For example, if you take pentapeptides, there will be, they treat that maybe for example 5 or 6 pentapeptides, minimum 5 pentapeptides, the same, then you can say that these two sequences which are have the identity of about 85%. And the second aspect is when they reduce the word size, then the efficiency decreases, right. Why? Because if you have the dipeptides or tripeptides, there is a possibility of several peptides, but they are not similar. Even then you can see several tripeptides or dipeptides, they are similar to each other, the possibility is very high. Even if you restrict the number of times, right, you can see the dipeptide or tripeptides. So, they started with the longer segments and they reduce the segments and increase the number of segments to identify the residues which are similar or redundant with each other. So, there is a compared size and another one is the number of same words, right, to identify the sequence identity. There are two conditions, one is the word size and the second one the number of same words, how many times the same words appear different at different locations. So, how the CD hit works, right? It uses the clustering techniques, right? So, one of the popular techniques that is called the k-means clustering. How the k-means clustering works? It is a method of cluster analysis which aims to partition n observations, right. For example, you have n sequences, right, n observations into limited number of clusters. For example, here k clusters, say 5 clusters or 10 clusters, in which each observation belongs to the cluster with the nearest mean, right, because that should be within this nearest mean. For example, if we have n observations like x1, x2, x2 up to xn, right, the k-means clustering, right, tries to put these n observations into k sets like different sets, so that it minimize the within cluster sum of squares, right. If you see the within the clusters, right, you can see the mean and any new sequence, right, the distance between these two features, right, they should make its minimum. So, you take any clusters, all the representative sequences within the clusters, right, that should be the minimum, fine. So, here I show an example. Here they like to have 3 different clusters. For any sequence, they try to find out the most probable cluster, and then once it is find with one cluster, then they reorganize, change the values, then again assign the other residues, so that for each cluster the value should be minimum. So, finally, you can see here is one cluster, here this another cluster, and put the third cluster. So, here I show one animation. So, here we have n data, n observations. So, we need to decide how many clusters you want to form. In this figure, right, how many clusters are desired? 5, right. Here k equal to 5, right, 5 clusters. So, first randomly they put into 5 clusters and try to see the average values for these 5 clusters and try to rearrange themselves, so that you can see for any specific clusters the mean value that is should be the minimum within that particular cluster. So, you can see a cluster here and you can see the cluster here, right. So, there will see 5 different clusters, right. We can see the different iterations, right, how these points moves. You can see the 5 stars, right, move everything together, and when we converge, you can see the clusters are very restricted, right, the deviation is very restricted. So, they can form any particular clusters. Then we take the representatives from each clusters, right, so that you can form any non-redundant data, right, in sequences or any data. So, what are the various features? How to get these sequences in a particular cluster? There are several ways, there are several features you can think of to cluster these sequences. So, one is a Hamming distance, right, based on the composition. For example, if you deal with sequences, we discuss with various features, right. What are various features we discussed for the amino acid sequences? Occurrence, composition, pair preference, molecular weight, hydrophobicity. So, any features, right, you can calculate. For example, if you take any property, say hydrophobicity, if you have 100 sequences, you can calculate the hydrophobicity for all the 100 sequences and make some threshold, right. For example, it is between 10 and 12 kilocal or 12 and 14 kilocal, 14 and 16 kilocal, right. Within that clusters you can put together. So, how many sequences do you require? So, you can put these different clusters, right, and then see what is average values. Then if you add or remove anything, so how this value changes, and if it is standardized, then you can get the representatives from each clusters.